So respiratory infections, which are the most common infections in humans, and out of them, the viral respiratory tract infections are the most common ones. And mostly they are upper respiratory tract infections, about which we have already talked in previous lectures. And we discussed a lot of viruses also. All right. So today we are going to discuss the lower respiratory tract infections caused by viruses. Viruses, right? So, what is viral pneumonia? It is a disease. Normally, the viruses they cause infection of upper respiratory tract infection, but when this infection reaches to the lower respiratory tract, to the alveoli, and it causes problem with the gas exchange and causes gas exchange abnormalities, then we call it pneumonia. Pneumonia, and it is because of the associated inflammation of the lung. And when this happens, the same can be seen in your X-ray and CT, etc. In your imaging, you will see imaging abnormalities. But the clinical picture, even with viral pneumonia, is not always same, and it depends upon what is the causative organism. It depends upon what is the age of the patient and what is the immune status of the patient. And as a result of all these factors, you will see a very varied picture of the pneumonia. Normally, viral pneumonias, they are much far less severe than the bacterial pneumonia. Yes, because bacterial pneumonia, they are a really severe disease. They are really toxic. When a patient suffers from pneumonia from a bacterial cause, he is really toxic and sick and bedridden. But most of the time, the viral pneumonias are not severe. Not severe. But some viruses, they causes severe pneumonia also, like severe acute respiratory syndrome viruses, coronaviruses, SARS-1, SARS-2, Middle East respiratory syndrome virus, which is also a coronavirus, and avian influenza virus or North American hantaviruses. These are known to cause very severe pneumonia. What happens in pneumonia? In viral pneumonia, the primary target is pneumocytes. Pneumocytes, these are the alveolar cells, type 1, type 2. So these are type 1, type 2 alveolar cells. These are pneumocytes, and they are the primary target of the viruses, and they cause diffuse alveolar damage. And the submucosa, the alveolar submucosa, becomes hyperemic. There are focal hemorrhages, edema, and then cell infiltrates into these alveolar space, interalveolar space, and which is associated with neutrophil mononuclear cells and the fibrin production. In advanced stage of the infection, there is fibrocellular intraalveolar organization. There is presence of histiocytes and multinucleated pneumocytes, which result into the fibrosis of lung. Causative agents for lower respiratory tract infections are influenza virus, parainfluenza virus, respiratory syncytial virus, rhinovirus, coronavirus, metapneumovirus, and among DNA viruses, adenoviruses, herpes simplex virus, varicella zoster virus, or cytomegalovirus. Most common infection, most common viral pneumonia in adult is caused by influenza viruses, influenza A and influenza B, and in children, most common agent is respiratory syncytial virus. Respiratory syncytial virus. So in children, the most common cause of viral pneumonia is respiratory syncytial virus, or we call it RSV, and in adult, the most common cause are influenza viruses A and B, right? And of course, that is about we are talking about the non-pandemic time. That is in non-pandemic time. When there is a pandemic, then of course the virus which is causing pandemic or even epidemic, or even a outbreak, local outbreak. That will be the most common cause. If there is a local outbreak of any of the virus, for example, there is a local outbreak of adenovirus, the first virus that should come to your mind should be adenovirus if there is an outbreak going on. But normally, the most common cause of pneumonia in adult is influenza virus, and in children, it is a respiratory syncytial virus. And these viruses are also an important cause. Rhinovirus, in some countries, it is the most common cause. For example, in Greece, the most common cause of virus pneumonia is, in some studies, it has been shown to be rhinovirus. So infection occurs. Where does this virus come from? It can come from three routes. One is the direct inoculation, that is the respiratory route. It has come to the upper respiratory tract, and then it goes to the lower respiratory tract, like influenza and RSV. Sometimes the infection occurs from the upper respiratory tract. For example, measles, it is an upper respiratory tract infection, but eventually the virus reaches to the lower respiratory tract by a contiguous transmission. Sometimes the virus comes from the hematogenous route, for example, cytomegalovirus. So these can be the routes. Most common and most important one is this, the respiratory route, direct inoculation through respiratory droplets. Kartikeya is saying I'm not audible. Am I not audible now? Am I audible? Okay, thank you so much. And immune response is both in pneumonia. It is both humoral and cell-mediated immunity. Humoral immunity are present in the respiratory epithelial lining, which are neutralizing antibodies, and cell-mediated immunity is mediated by both T lymphocytes, CD4 T lymphocytes and CD8 T lymphocytes. CD4 T lymphocytes, which attract immune cells in MHC class 2 response. CD8 T lymphocytes, they induce an MHC1 response, which is the lysis of the infected cells and apoptosis of the infected cells. Now, coming to the influenza virus, this will be a little bit of recapitulation of your previous lecture, upper respiratory tract infection, because we discussed influenza virus there also, and some aspects which we didn't cover at that time that we will be discuss now. It is a member of myxovirus, orthomyxovirus. Influenza virus is orthomyxovirus. These parainfluenza, mumps, measles, RSV, they are paramyxoviruses. And mixa has come from their heme agglutination ability. They have a surface protein called heme agglutinin, and because of that, they can agglutinate RBCs. The term mixa has come from there. The influenza virus causes infection by airborne droplets, and attachment is mediated by heme agglutinin, which is a protein on the surface of the virus. And there is also another protein neuraminidase, and which liquefies the receptor, the mucus, and it breaks the receptor and helps the virus in releasing from the cell. The antigenic structure of influenza virus, one is ribonucleoprotein. On the basis of ribonucleoprotein, we divide them into three types: A, B, C. Type A causes infection in human, animal, birds. Type B and C cause infection in humans only. In type C, there are 7 segments. Otherwise, normally there are 8 segments, which lacks neuraminidase. In fact, many of the paramyxoviruses, they lack this neuraminidase activity, and some have only heme agglutinin activity. So primarily, the virus needs heme agglutinin because that is the protein that will help in the attachment. Heme agglutinin and neuraminidase. hemagglutinin helps in the attachment, neuraminidase helps in the release of the virus from the cell. And the genome of the influenza virus is RNA, segmented RNA, and there are 8 segments in influenza type A and B and 7 segments in type C. And because of these segments, because gene for H, hemagglutinin, and neuraminidase, they are present on different segments. So one virus can have a different hemagglutinin, another neuraminidase. So in human influenza viruses, there are 4 types of hemagglutinin are found: H1, H2, H3, and H5. And in human, only 2 types of neuraminidase are found: N1 and N2. N1 and N2. So in human influenza viruses causing human infection, 4 types of hemagglutinin are present and 2 types of neuraminidase are present: N1, N2, H1, H2, H3, H5. Otherwise, there are up to 16 hemagglutinins and up to 9 neuraminidase, which including which causes infection in other animals, birds, and swine, etc. This was the nomenclature structure of the influenza virus we talked about earlier. The very important aspect of the genome of influenza virus is that it keeps on changing by 2 methods. One is antigenic shift and other is antigenic drift. Antigenic shift is because of the segmented property of the genome, because it happens because the genome is segmented. RNA of the influenza virus is segmented, and because of that segmented property of the genome, there can be an antigenic shift. And antigenic shift is because of reassortment of different segments of the RNA. When one animal is infected with 2 types of influenza viruses, so when a single cell, 2 types of RNA viruses are present, so some segments can carry over from a new variant can come with some segments of this virus and some segments of this virus, right? Normally what happens, these segments, they randomly go in one virus or in other virus, so only that virus will survive which is compatible for the survival. If, for example, in one virus only one type of segments are there, that virus will not survive. So a compatible virus which has all the necessary genes present in it will only survive, and this can result into a new hybrid virus. So this is antigenic shift. Antigenic drift is our normal mutation, which can occur in any type of RNA viruses, and it is more common in RNA viruses because of the RNA property of RNA polymerase enzyme, lack of proofreading activity of RNA polymerase enzyme. It results into antigenic drift. Drift means slowly the virus is changing over time because of mutation. Antigenic shifts are sudden, they appear suddenly out of the blue. So because of this antigenic shift and antigenic drift, the influenza virus keeps on changing. And with this change, what does the virus achieves is that it can infect more people who are not immunized for that antigen. Because infection of the influenza virus provides immunity, but if the virus has changed its antigen, it can now cause infection into the population. In this picture, if you see, the horizontal line is representing the drift, and the vertical lines, they are representing the shift, antigenic shift. So in 1918, the virus came into existence, that what H1N1, and it kept on changing slowly, and because of the antigenic drift. And in 1957, because of antigenic shift, it became H2N2, and it kept on changing slowly with antigenic drift mutations. And then because of the antigenic shift, this H2 changed into H3, and like that the virus keep on changing over the time, both because of antigenic drift and also because of antigenic shift. So in 1997 it became H5N1, in 1999 it became H9N2, in 2003 H7N7, H5N1, and in 2009 the swine flu pandemic that was caused by H1N1. So this is the real importance of the antigenic shift and antigenic drift in influenza virus infection. Now coming to the avian influenza, which is H5N1. It causes infection in birds, particularly the wild birds, and in fact, in wild birds, it is commonly present in their saliva, nasal secretions, intestine, without causing them any disease. But when this influenza A virus, avian influenza A virus, it comes in contact with the domestic birds, it causes infection in domestic birds. Right. So somebody is asking a question. I'll answer that after finishing the influenza part. So avian influenza A, it normally causes infection in birds, and it is present in wild birds, and it can cause infection in domestic birds. And because it is very contagious, and it can cause infection in domestic birds, and there are two forms of in birds, that is, one is low pathogenic and the other is high pathogenic. Low pathogenic just cause like a mild infection, such ruffled feathers and drop in egg production. High pathogenic form, it causes more rapid infection, and it is very infectious also. Besides, it has very high mortality rate, and it can reach up to 90 to 100% mortality within 48 hours. That is, we are talking about the birds only. In humans, till now, 503 reported cases are there by WHO, with 299 deaths. And most cases, they result only to the people who came in contact with poultry farms, right? And the risk of avian influenza is generally considered to be low. And because these viruses, they are not adapted to cause infection in humans, and human to human transmission is not known, or it's really very rare. So then what is the problem? Why do every time there is avian influenza outbreak among birds, uh, health officials, they start panicking? The real concern is that one is H5N1 virus has a potential to cause pandemic. Why? Because the population is not immune for this virus. Somehow the virus is not infecting too much to humans. It is not very infective. There is very less or rare or unknown human to human transmission. But the potential of this virus is really big. And besides, it is resistant to amantadine, drementadine, which are the M2 blocker drugs for influenza virus. Uh, it has caused different outbreaks in Hong Kong, Southeast Asia, Canada, Egypt. And if you look at the epidemiology or the toll of these diseases, if you see in Hong Kong in 1997, there were 18 cases, 6 were fatal, 18 cases with 6 deaths. In Vietnam, 23 cases, 16 deaths. In Thailand, 5 cases, 4 deaths. In Cambodia, 6 cases, all 6 died. In China, 18 cases, 12 died. So if you see, number of cases are really very small, but the percentage of deaths, the fatality is really high. It's more than the coronavirus, it's more than MERS, it's more than SARS-1 and SARS-2. Although the number of cases are very low, but the mortality is highest among all the respiratory viruses, including coronavirus, including the current ongoing coronavirus pandemic. So that is a very big concern. So it can cause a possible next pandemic, uh, and at present it is present in birds. More than 200 human cases have been reported, more than 500, and with 200 around deaths. Incubation period is 2 days. Uh, Route of infection is respiratory route. Source of infection is infected patient. Transmission, contaminated hand, fomite, aerosol, that is for all influenza viruses. And complication can result into primary pneumonia, secondary bacterial pneumonia, encephalopathy, transverse myelitis, myocarditis, pericarditis, Ray's syndrome, etc. Another variant is H1N1, which is also known as swine flu. This is the same variant which caused the huge pandemic in 1918, 100 years from now, and it resulted into, I mean, millions of deaths. Some around 50 million deaths. The same H1N1 strain caused a pandemic in 2009, and this new strain of influenza virus was identified in 2009, and it was commonly referred to as swine flu. And it was the product of the reassortment of 4 strains of influenza A virus: one human type, one bird type, and 2 pig type, which means it was a result at some point of time. There were 4 different influenza viruses in one cell. And because of the reassortment in the 4 viruses, this strain came in 2009, and it caused a pandemic and much uh, panic also around the world. And so far in India also, 34,000 confirmed cases have been reported. The drug of choice is oseltamivir and zanamivir, which are neuraminidase inhibitor. Diagnosis of influenza viruses, of all influenza viruses, is same. That is the throat swab. Among today we are talking about lower respiratory tract infections, uh, but the sample collection in lower respiratory tract infection, particularly the viral respiratory tract infection, most commonly collected sample is throat swab, which is the upper respiratory tract sample. But because virus is also present in throat if there is pneumonia, so we collect that sample, and it has a good sensitivity, a fair good sensitivity, around 85 to 90% sensitivity. But if you will ask me what would be the ideal sample, because we are dealing with the lower respiratory tract infection, so ideal sample would be a bronchoalveolar lavage or a lung biopsy or a transtracheal aspirations. But because they are difficult to collect, the routine sample for all viral pneumonias, be it influenza or be it coronavirus or any other virus, the sample of choice which is commonly collected is throat swab. Other samples which can also be collected easily: nasal swab, nasopharyngeal swab, aspirates, washings. In fact, in coronaviruses, we started with 2 swabs, a throat swab and a nasopharyngeal swab. And the sample should be transported with cold chain maintenance in a viral transport medium, and it should be chilled but not freeze during the transportation. And for influenza virus, the diagnosis, gold standard for diagnosis is culture. Culture is done in embryonated chicken eggs, 9 to 11-year old, in the allantoic cavity of the egg. And allantoic fluid is harvested after a few days, 4 to 5 days, and then we look for heme agglutination titration, viral titration. If it comes positive, it means it is influenza virus. Apart from egg culture, we can also do cell culture, primary monkey kidney cell culture, Medin Derby kidney canine cell culture, CPE. We will look for cytopathogenic effect in 2 weeks, syncytia formation or cytoplasmic basophilic inclusion bodies or heme adsorption. Some antigen tests are also available, rapid antigen detection test. We take the sample, extract the antigen, and we look for the presence of antigen. Most commonly used tests for the diagnosis are the molecular methods, RT PCR. In case of influenza, we or respiratory syncytial virus or other paramyxoviruses, we do RT PCR, and we do, first of all, we look for whether it is influenza A or B. Then we do the RT PCR for the individual hemagglutinin. We look for whether it is H1 or H2, like that, and with the help of that we can make the diagnosis whether it is H1N1 swine flu or H5N1 avian influenza or some other strain. It is the preferred method because it gives the diagnosis in a matter of hours, and it is also very sensitive. Treatment for influenza, there are two types of drugs available. One are M2 channel blockers, amantadine, remantadine. They block M2 ion channel, which are required for uncoating within 48 hours of illness. The other are neuraminidase inhibitor, oseltamivir, zanamivir, premavir. They prevent relapse and spread. These antiviral drugs, they are best, they give best results if they are given within 48 hours of infection. Neuraminidase inhibitors, they act both on influenza A and B. M2, they act only on influenza A. Now coming to the prophylaxis for influenza virus, chemoprophylaxis can be given amantadine, remantadine, and vaccine. Three types of vaccines are available. Inactivated vaccine, they are grown in, viruses grown in chick embryo and inactivated by formalin. And the strain that we give for influenza vaccination, we give multiple strain, 2 or 3 strain. One is which is the present, which is causing the present outbreak, or which is causing infection at present time, and the another strain which we are expecting to cause infection in future, in the next year. So like that, we add multiple strain of influenza virus in vaccine, so that multiple strain of influenza can be covered. And the vaccination of influenza virus is given every 2 years, because the older strain, antibodies against the older strain will not be protective for the newer strain. Live attenuated virus, it is given as a nasal spray. It is available since 2003, and it is live attenuated, which is attenuated by serial egg passage, but it is temperature sensitive. So, and a recombinant vaccine, which is also under evaluation. Right. So this was about the influenza viruses. There was a question. Let me. Shashwat Aran asked me question, sir, can antigenic shift and drift could also lead to large pandemics just like the current one? Antigenic shift can lead to the pandemics, current pandemics. Drift does not cause pandemics. Drift causes the seasonal variation of the influenza incidence. It is the antigenic shift that causes large pandemics. Because of the antigenic shift, the virus is completely changed, and whole the population of the world is not immune to that new virus, and because of that, it results into the pandemic. Now you are asking, Vishwa is asking another question about corona. I'll discuss that when we will be talking about coronavirus. So this was about influenza virus. Another myxovirus, paramyxoviruses they cause infection similar to influenza, but a milder form of it, and we call it paraflu. Paraflu caused by parainfluenza virus. There are 4 types, 1, 2, 3, 4. Type 4 has subtypes A and B. Type 3 is the most prevalent of them. Infection occurs worldwide, usually endemic. Primarily in young children, reinfection is common. It can cause upper respiratory tract infection as well as lower respiratory tract infection. Like diagnosis is just like influenza virus by RT PCR. Vaccine is not available against parainfluenza virus. Now coming to respiratory syncytial virus, which I said is the most common cause of lower respiratory tract infection among children. It causes common cold, cause pneumonia in elderly, and febrile rhinitis and pharyngitis, bronchiolitis, pneumonia, and sudden infant death syndrome. Infection is just like influenza, by droplets, direct contact. In RSV pneumonia, there is bronchitis and bronchiolitis, which is associated with necrotic mucosal lesion and mucus plugging of the respiratory tract. Host defense is because it's a viral infection, both humoral and cell-mediated immunity is required. Diagnosis is by cell culture or antibody detection or by RT PCR. Earlier, the diagnosis of virus was used to be done either by cell culture virus if a virus is culturable, or by the antibody detection. Nowadays, because RT-PCR facility is available, so most of the viral infections are best diagnosed by RT-PCR or PCR if it is a DNA virus. Controlled prophylaxis, there is no vaccine is available, but in some countries, passive immunization is given for the control of respiratory syncytial virus. The preformed immunoglobulins are given for the control of respiratory syncytial virus. Treatment is supportive care. Ribavirin is administered by inhalation. Another paramyxovirus is metapneumovirus. There are two types of metapneumovirus: avian metapneumovirus and human metapneumovirus. Human metapneumovirus is similar to one type of avian metapneumovirus. So it is thought that this human metapneumovirus is it is a derivative of avian metapneumovirus. It causes upper respiratory tract infection. It is a cause of upper respiratory tract infection in 5 to 15% of cases, and lower respiratory tract infection in 5 to 15% of cases. Diagnosis will be similar: cell culture and RT-PCR. Right. Antiviral drug ribavirin is drug of choice for metapneumovirus. Now coming to the coronaviruses. Coronaviruses, they cause respiratory tract infection, and they vary from a common cold to severe pneumonia. Coronaviruses were discovered in 1937 in chickens, and at that time it was known that they cause infection only in chickens. The first time they were discovered in humans to cause infection in human, it was in 1965. So first time human coronavirus was isolated in 1965, and it used to cause it at that time. It was known that they cause only common cold and no severe infection. They were a common cause of common cold. And from 1965 till 2002, they remain only a cause of common cold. They are classified into the classification of coronavirus. They belong to family Coronaviridae, genus Coronavirus. They have a non-segmented single-stranded RNA. Till 2002, there were known only two human coronavirus 229E and OC43, and they used to cause only common cold. But first time in 2002, a new coronavirus came into existence. It came from some animal or etc. in China in 2002, November 2002, and then it rapidly spread to different countries: Hong Kong, Canada, Taiwan, Philippines, and it caused severe pneumonia, and it was really very infective. Severe pneumonia. The name SARS has come from severe acute respiratory syndrome. This was the first time when a coronavirus caused a severe respiratory tract infection, and that ended into a pandemic, epidemic. And this spread very fast to many countries, and it resulted into the death of some many people in some 14 to 15 countries. After 2000, but it was very rapidly contained in, and it did not become a pandemic like that. In 2012, another coronavirus was identified in Middle East, and that outbreak is still going on since 2012 until today. There have been around 2,519 cases with 866 deaths. So mortality of MERS coronavirus is around 34%. 34%. The mortality in SARS coronavirus is around 15%. 15%. And in 2009, another variant of coronavirus, SARS coronavirus, was discovered in China, Wuhan, China, and till today it has caused 17 crore cases with 38 lakh deaths. So 17 crore cases and 38 lakh deaths worldwide. There are 4 genera of coronaviruses: alpha, beta, gamma, and delta. Alpha, beta, gamma, and delta. All 3 of the severe infection causing coronaviruses, they belong to the beta group. SARS-CoV, the first SARS, the MERS, and SARS-CoV-2, they all belong to the beta group. There are total 7 coronaviruses known to cause infection in humans. 3 SARS coronaviruses and 4 human coronaviruses 229, NL63, human coronavirus OC43, human coronavirus HKU1. So 1, 2, 3, 4, 5, 6, and the newer SARS-CoV-2. 7 coronaviruses, including SARS-CoV-2. Coronavirus, the name coronavirus has come from the spikes, because corona means crown, and because of these spikes it looks like a crown in electron microscopy, so name has come from there. There are the genome of the SARS-CoV-2 virus is RNA, single-stranded RNA, and a nucleocapsid protein is present with RNA, and there is an envelope. Envelope has got these spike proteins, and this membrane has membrane protein. The genome is a non-segmented RNA. It has 5 cap and 3 poly A tail, which is common with all RNA viruses. They have a 5 cap and poly A, 3 prime poly A cap. Size of the genome is around 30,000 base pair, which is 30 kb. The viron is about 22 nanometer in diameter. This is the whole genome, and the small part of the genome is the one which codes for the structural protein, and the large part of the genome is for non-structural protein, ORF1A, ORF1B. ORF means open reading frames. The small part of the genome which codes for structural protein, it codes for spike protein, envelope protein, membrane protein, and nucleocapsid protein. So there are 4 structural protein: N, S, M, E. N is the nucleocapsid, S is spike, M membrane, E is the envelope protein. There are 16 non-structural proteins. The envelope is a lipoprotein, host drive lipid. Spike protein is the one which helps in the attachment of the virus to the host cell. So this is the spike protein which acts as a receptor to bind on the host cell. And on host cells, the receptor is ACE2, angiotensin converting enzyme, which is an enzyme which converts into angiotensin 1 into angiotensin 2. That enzyme acts as a receptor for the virus. So this is the spike protein and ACE2. They together bind, and then the virus enters into the cell. And where these ACE receptors are present. They are present in nasopharyngeal mucosa, oral mucosa, heart, lung, colon. ACE receptors. They are present throughout the respiratory tract and in some intestine also. So this ACE receptor, ACE2 receptor, it helps binding the coronavirus to the surface, and then the virus enter into the cell and the RNA is released into the cytoplasm. Then the cycle of the replication goes on. So host cell entry is mediated with the spike protein of the virus, which binds to the angiotensin converting enzyme receptor for SARS-CoV-2. So what happens after the viral entry? There will be activation of the macrophages, and the macrophages will release pro-inflammatory cytokines, IL-6, IL-10, IL-2, IL-1, TNF alpha, interferon gamma, etc. And together they will cause acute lung injury, and it will result into the respiratory failure. The natural host of coronavirus is civets or raccoons or pangolins, and from there the human infection, the SARS-CoV-2 virus resembles more to the bat coronaviruses. So it is expected that the SARS-CoV-2 virus has come from the bats, either directly from bats to humans or through another intermediate host. It can be a dog, it can be a snake, it can be a pangolin, but we do not know for sure what was the nature of infection to the human. The infection is transmitted by the spread of the droplets shed in the respiratory secretion. A viral load is peaked around 10 days and 13 to 14 days in stool. SARS viruses seem to enter into the body through eyes, nose, mouth, and touching the objects that are contaminated with virus. Pre-symptomatic and asymptomatic transmission is also seen. So the transmission is not only by the cases, pre-symptomatic cases and asymptomatic cases, they can also transmit the infection. And pre-symptomatic means 1 to 3 days before the onset of illness, the patient is perfectly fine, he has no symptoms. Symptoms are yet to develop, but still he can transmit infection to other. And some people, they are completely asymptomatic, they do not develop symptom at all, but still they transmit infection to others. And symptomatic cases, they can obviously can cause infection too. Let me see. Okay. Clinical manifestation, incubation period is 4 to 5 days. It can last up to 14 days. So incubation period, routinely, most common type is 4 to 5 days, but it can be up to 14 days. Fever, chills, cough, shortness of breath or difficulty in breathing, fatigue, muscle or body aches, headache, new loss of taste and smell, sore throat, congestion or running nose, nausea, vomiting, diarrhea, dermatologic manifestation. These are the frequent symptoms. They can be present they can be present, they cannot be present, few of them can be present, or all of them can be present. So in different people, different presentations are seen. There are some pictures. Okay. So initially there is a sudden onset of high fever with dry cough, then it end up into chills and shiver and muscle aches, and after some time there is difficulty in breathing after 3 to 4 days, from the onset of symptoms, and then there happens severe pneumonia. So this is the routine pattern of the disease, but some people can have infection only up to this stage, other people can have infection only up to this stage. Very few people go to the severe stage. So mild to moderate, in 81% of the cases the infection is mild to moderate. Severe infection is seen in 14% of the cases, and only 5% of the cases are the critical ones. What is mild, what is severe, and what is moderate? Mild means only fever or with some symptoms without dyspnea, without respiratory difficulty. Mild to moderate respiratory illness means temperature, fever with some respiratory difficulty. And severe respiratory illness means patient has radiographic evidence of pneumonia, acute respiratory distress syndrome. Autopsy findings are consistent with pneumonia, sepsis, septic shock. That is severe respiratory illness. And the onset of the illness is like dyspnea develops in 5 to 8 days, acute respiratory distress syndrome that develops 8 to 12 days, and ICU admission, the mean time of ICU admission is 9.5 to 12 days. Risk factor for the severe illness, advancing age is the most important risk factor, because it has been seen that mortality is 14% in people who are above 80 years of age, 8% in 70 to 79%, 3.6% in 60 to 69%, and 1.3% in 50 to 59%. So as the age is decreasing, mortality is also decreasing. So highest mortality is in above 80 years of people. So advancing age is most important risk factor for the severe illness. And comorbidities like underlying cardiovascular disease, diabetes, chronic respiratory disease, cancer, they are also a very important risk factor for severe disease. Some people are raising hands. Let me first see that. Mm, okay, it's nothing. Some people have just entered their roll number. Okay, I know you are there. Thank you so much. Okay, mutation and variants of coronaviruses, like all viruses, because coronavirus is RNA virus, so it causes the virus that causes COVID-19. It also changes over time. Over time, in fact, all the viruses they change over time, particularly the RNA viruses. But time, I mean, for viruses, time means something else. Uh, they do not have a watch or they do not have a calendar. For them, time means replication, how many times they are replicating. So normally all the viruses, RNA viruses, they change over time. But if there is a pandemic or there is a lot of infection going on, lot of replication is going on, so rate of changing also increases. So the rate of changing is also increases because of the rapid infection, rapid replication of the virus. Otherwise, it is not a new thing for RNA virus to change over time. So like that, coronavirus is also changing because there is so much infection going on. So the virus is also changing rapidly. Rapidly means in our time frame, they are changing rapidly. But otherwise, the rate of change, if you see per replication, is similar to other RNA viruses. So most changes, most changes in the RNA viruses, they are little or they have no impact for humans, because it is a routine change because of the lack of polymerase proofreading activity of the polymerase enzyme. So at some places the RNA is going to change, and most of the time these changes are of no consequence, no consequence. But sometimes they have consequences. It can the change new variant can have a high infectivity, it can be easy to spread, and it can cause more severe disease, or it can cause uh, it can become resistant to the vaccines. The performance of the vaccines can be diminished, or performance of the therapeutic medicines, although real known therapeutic medicines are not there. But particularly the monoclonal antibodies, which are not being used in India, but in some countries they are using monoclonal antibodies or about to use monoclonal antibodies. So if the virus changes, the monoclonal antibodies are not going to work for that changed virus. Another important aspect is the diagnostic tools, because we are doing diagnosis by RT PCR, and that RT PCR, it detects the RNA. So if that RNA is changed somehow, particularly in that place where the primer binds to the PCR primer binds, if that part is mutated, our diagnostic tool will not be able to diagnose the virus, or their sensitivity will decrease, and there can be other concerns. So normally the change in the virus does not cause much concern, but sometimes if the virus has changed and it is in causing these problems, so that becomes a concern. And these variants which have these properties, they are called variant of concern. This is a term coined by WHO for those variants which have either high infectivity or high severity, or vaccine performance is involved, or the therapeutic medicines involvement is involved, or the diagnostic tools are not able to diagnose that new variant. So that variant is called variant of concern. This is normally we do not uh, tell about the phylogenic of the virus. The virus that mutates, they form a family tree, just like a human family tree. The viruses, they also have a family tree, because there in a tree there is a space we can sit under the tree, but here we do not want to sit under the tree, so we have make it a round structure so that we can save the space uh, below the stem. Otherwise it is just a tree, a family tree. So one virus was strain was A. When it separated into two, it became B. Then B separated into two branches, they become B and B.1. When B.1 separated into two branches, it became B.1.1. Like that, there are many branches. So these lineages are called pango lineages. B.1.1, 1.1. This point shows a branch, a point of branching of the lineage. So it is very difficult to remember the virus on the basis of these lineages, because it is a very long lineage, 1.1, 2. So what did people do? They started to calling them by their name of origin, like the United Kingdom variants, South Africa variant, or Brazil variant, or the Indian variant. But that wasn't perceived as good practice, because it wasn't politically correct for many people. So WHO has given them name as alpha, beta, gamma, delta. The alpha is the one which is known as earlier as UK variant. Beta is the one which was earlier known as South Africa variant. Gamma is the one which is which was known as earlier as Brazil variant, and delta is the one which was known as earlier Indian variant. So the delta has now acquired a mutation of the Brazil variant, so that one is called delta plus. So we have these alpha, beta, gamma, delta. These are variants of concern. Otherwise, if you look in the population, the virus, there can be many more variants, but they are not of concern, because it is an RNA virus, so every one virus will be different from other virus. It's going to be different, because every replication is going to change the nucleic acid in some way. But for us, the important variants are these till now. If anything comes in future, that will be a different thing. Lab diagnosis. Whenever we talk about the lab diagnosis of coronavirus, this picture come to our mind. Lab diagnosis is again just like influenza virus. There is nothing different in the lab diagnosis of coronavirus from the influenza virus, apart from that the uh, viral culture you will do on different cell lines, the RT PCR you will do using different primers, and besides all the precautions you will keep in mind while collecting the samples. And that is also not specific for coronavirus, it is specific for the potential of the virus to cause pandemic. So if any new virus come, for example in 2019 swine flu virus came, the same pattern, same method was followed to for the sample collection, for the diagnosis for the virus. Sample is taken throat swab and nasal swab, and it is kept in a viral transport medium. Viral transport medium is nothing but a liquid which keeps the swab moist, because if it gets dry, the virus will die. And it has some antibiotics, etc., some buffer, so the virus, the environment of this is conducive for the virus. It doesn't get dry, the bacterial growth is not there, etc. Otherwise, if you want to do PCR, the RT PCR directly from the swab, you can also perform it. There is no need of RT PCR. If you, VTM is the need only for the transport of the swab. Otherwise, you can just mix the swab into a normal saline. You can also perform RT PCR. And because of the pandemic potential of the virus, it should be packed into a triple packaging system, one box with viral transport medium and swab, which is surrounded by some absorbent material, tissue paper. Then it is kept in a box, then another box. This is how the samples for respiratory viruses are transported. Swine flu, avian flu, coronavirus, all of them are transported like that. So sample is nasopharyngeal swab, throat swab. Other samples can be stool, blood, urine, etc. It should be transported maintaining cold chain. Laboratory diagnosis, antigen detection, antibody detection, virus isolation, molecular test detection. Molecular test detection are the most common one. They detect E envelope gene for envelope protein, RTRP, nucleoprotein, or ORF region. Real-time PCR has 80% sensitivity within 5 days of onset. So sample is collected in VTM, then we extract RNA from that sample, and then we put it on RT-PCR and we look for the fluorescence. If the sample is positive for coronavirus or for any other virus, this RT-PCR procedure is same, only the primers are different. So if it is positive, it will give a curve of fluorescence. Virus isolation is done on varicose embryonated monkey kidney cells. This is, first of all, it is not done for diagnosis. It is done only for research. And the second thing is, it can only be done in BSL-3 facility, biosafety level 3 facility. RT-PCR can be done in BSL-2 facility. So biosafety facilities have their gradation, biosafety level 1, 2, 3. So culture can be done in BSL-3, RT-PCR can be done in BSL-2. Antigen detection, nucleoprotein or M antigen, they are detected, but the sensitivity is not very good. Antibody detection can be done, but for the diagnosis of viral infection, it is of no use. It is done for these serosurveillance studies. Only for serosurveillance studies can be done by ELISA or immunofluorescence, etc. Other laboratory findings, lymphopenia, 83% of the hospitalized patient, elevated serum alanine aminotransferase, AST, lactates, dehydrogenase C-reactive protein, high ferritin is associated with greater illness severity. Elevated D-dimer is associated with mortality, and high plasma inflammatory markers, they are the marker for immune dysregulation. Because it is said the main pathology in infection is the immune dysregulation instead of virus causing the lysis of pneumocytes. Radiographic findings, bilateral airspace consolidation, computerized tomography, bacterial peripheral ground glass opacities, like this you see in CT. Clinical management and treatment is important for infection prevention and control measures, and supportive care is given, supplementation of oxygen and mechanical ventilation when required. If there is no hypoxia, so no hospitalization is not required. The disease can be managed if it is mild at home. For moderate infection, severe infection, hospitalization is required for the supportive management of the for the supportive management and the management of the complications. Complications are pneumonia, hypoxic respiratory failure, sepsis, septic shock, cardiomyopathy, arrhythmia, acute kidney injury, and complication from prolonged hospitalization, because prolonged hospitalization itself causes a different problems, and secondary bacterial or fungal infections. For them, you need to hospitalize the patient. Treatment is isolation, supportive care, broad spectrum antibiotics, and antiviral. Currently used antiviral drugs are remdesivir, favipiravir. Remdesivir is a adenosine analog that incorporated into the viral nucleic acid. When the viral nucleic acid is replicating, this remdesivir, because it is adenosine analog, it will bind to the nucleic acid and it will block the chain. Favipiravir, it inhibits the RNA-dependent RNA polymerase. They were very widely used, but the effect of them is not proven by uh, the control studies. Corticosteroids are given based on the pathophysiology of the disease, because main pathophysiology is the immune dysregulation. So steroids are given because of that. Initially, hydrochloroquine was given to the people, but the role of hydrochloroquine is also not proven. Azithromycin is given also to prevent the secondary infections. Complication of COVID-19, acute respiratory failure, pneumonia, acute respiratory distress syndrome, acute liver injury, cardiac injury, secondary infection, acute kidney injury, septic shock, fibrosis, and multi-organ failure. Antiviral drugs and vaccines. Now coming to the vaccines, luckily we have vaccines available now for coronavirus. There are 3 types of vaccines: RNA vaccine, adenovirus vector vaccine, and inactivated virus vaccine. Adenovirus vector vaccines, in which animal source adenovirus is taken, and a small part of SARS-CoV-2 RNA is incorporated into the adenovirus, the animal origin or the chimpanzee adenovirus. And because of that uh, genetically modified uh, RNA, because this adenovirus now contains SARS-CoV-2 RNA in this, this will also make this spike protein on its surface. So this adenovirus can now be given as a vaccine agent. So currently, the Oxford AstraZeneca vaccine, which is known in India as Covishield, is a adenovirus vector vaccine. Russian Sputnik COVID-19 vaccine is also adenovirus vector vaccine. Covidicea, Johnson and Johnson COVID-19 vaccine, they all are adenovirus vector vaccine, in which in one adenovirus we have genetically engineered a small part of SARS-CoV-2 virus RNA for spike protein, and they also make spike. The adenovirus also make the spike protein on its surface. The inactivated virus vaccines are the conventional virus vaccine, in which we grow the virus and kill the virus, and we use it after purifying. We use it as a vaccine. The Covaxin in India is an inactivated virus vaccine. CoronaVac in China is an inactivated corona vaccine. Covivac in Russia is. These all are inactivated virus vaccines. The most routine technology for making the viruses is using a killed vaccine, inactivating by formalin. RNA vaccines. RNA vaccines, they were in pipeline for many infections for a very long time, but they were never used for any disease. In coronavirus, after the coronavirus pandemic, the RNA vaccines, they are now available for the first time in human history for a virus, that is coronavirus. The Pfizer BioNTech COVID-19 vaccine or Moderna COVID-19 vaccine, they are RNA vaccine, in which we make the RNA in vitro, and this RNA into a lipid nanoparticle is used as a vaccine, because RNA itself cannot survive in body, because there are many enzymes which will degrade the RNA. So this RNA is given as a combination with lipid nanoparticles. These lipid nanoparticles, they surround this RNA. RNA. So this RNA, when it will enter into the tissue, it will be taken up by the cells, and when it will reach into the cytoplasm, this will act as a messenger RNA, and it will translate the protein, and that protein will appear on the surface of the cell, and that will act as immuno antigen for the development of immunity. Other things, hand hygiene, personal protective equipment, environmental cleaning, social distancing, respiratory hygiene, and cough etiquette, social distancing, including the lockdown, etc. They are used for the prevention of the infection. That is about the coronaviruses. So if you have some questions, you can ask. Vishwa Rakesh had asked this question: did incorrect sample choice lead to false negative in corona? Yes,